Well, good afternoon, queridas y queridos. Welcome to our new edition of EcoEncontros. Today I have the pleasure to present Marco Antonio Chávez-Tinoco. He is currently doing a PhD in plant biotechnology at the Center for Research and Advanced Studies of the National Polytechnic Institute in Mexico. Marco also has a master in sustainable agricultural production from the same university, from the National Polytechnic Institute of Mexico. And he's been studying fauna in transformed landscape. And today he's going to present his the work that he did during his master's. He works with exotic species control at a lake in Mexico. So Marco, thank you very much for your disposition to give this talk. So you might start now.
Muito obrigado pela apresentação. Estoy muy contento por poder presentar un poco de lo que hacemos en México. Perdón por mi portuñol. Sorry for my bad Portuguese. Well, thank you for your attention. Control of exotic species and monitoring of water quality in Western Mexico, a long way to go. And we're going to focus in Mexico because, well, Mexico is one of the countries with the largest water resources in Latin America. So our history is marked by the presence of lakes and rivers, as in the case of Tenochtitlan and Pátzcuaro, who were important cities in the center of Mexico during the pre-Hispanic time. However, these resources currently face several challenges. The greatest of these challenges is the loss of biodiversity. It is important to know that Mexico has an enormous wealth of endemic species, of which approximately 500 have been described only in the water environment. This diversity is driven by the introduction of invasive species, which displace native ones, extreme environmental changes, habitat destruction, and even overexploitation. In the particular case of the introduction of invasive species, their scope is wide-ranging, as they tend to occupy ecological niches without direct competition, leading to a competitive exclusion of native ones, as well as cause specific environmental damage. For example, in the case of Plecostomus, these fish cause erosion of the banks of lakes and rivers to their particular reproductive habits. Finally, it has been shown that the food chains are being restructured with the introduction of these foreign species. The research in which I will delve further is derived from a project in which I was involved since my thesis degree and my master's degree, as well as in the time between master's and doctorate in the last year, and from which we have submitted an article in the past weeks. The project was led by my advisor, Carlos Escalera Gallardo, and Rodrigo Moncayo Estrada in the Laboratory of Aquatic Ecology in Cidir, Michoacán. We will focus in Lago La Alberca, which is located, which has with special environmental conditions. For example, geothermal activity. It is an endorheic lake that has no entrance beyond the springs that originated and the rains of a storm. The climate is warm, with rains in summer and winter, and a maximum depth of 36 meters. This lake has been proposed by our research group as a reserve for native species. Originally part of the main vessel of Chapala Lake, which is the largest lake in Mexico. However, the drying process of the middle of the last century for gaining new lands for agriculture in the region, this lake were isolated, and despite being about 100,000 times smaller, it was left with almost half of endemic species, with 11 species described at the moment. However, at the beginning of the first decade of 2000, several species were introduced by the government to promote sport fishing. The lobina and two species will serve as prey, Coptorendali and Lepomis macrochirus. Despite the high salinity of the environment of about 2 parts per million, was lethal to lovine, which prefers a quarter of that concentration. The disappearance of the predator made that Copton and Lepomis began to thrive into the environment, so much that, as we will see later, has become a problem. The ecological study we started then to know the degree of colonization of these invasive species was derived from that in 2016, in my degree thesis, we found that these two species were already the most important in terms of biomass, and we needed to make a comprehensive study to propose strategies of control and utilization. The lake was divided into 3 sampling zones, north, center, and south, in which the environmental values were recorded through the help of an hydrolab. Organisms were collected like benthic fauna, plankton, and fish. The samples were obtained in the morning and at noon. When the nets of fishes were taken out of the lake, the native ones were returned to water, and the invasive ones were conserved in ice and then fixed in alcohol for subsequent tests. First, we found that it is a polymictic lake with a uniform distribution of oxygen through all the column of water. With regard to this parameter, it's interesting that 2 of central and southern station present optimal oxygenation above of 5 parts per million, that is the necessary limit to preserve life in the environment. It is also a saline oligotrophic lake where the energy level is limited to support biological communities. In this case, we found that exists a zonification of the lake in the colonization of the species and the colonization especially in the ichthyic fauna. The surprising thing was that when we compare our samples with previous information from our group, we see a substantial increase of invasive species, as well as an evident decrease in species such as Chapalictis encaustos, Codea tripinis, Cenotoca baeata, and Zogonithecus polypechus, which as we shall see below, appears to have strong competition for food items. Contrasting this data with information collected in 2007 and 2008, we can see that the rise of invasive species was constant, and we can see that it was a staggered process. In this sense, we must remember that a great part of native species belonging to the genus Specilidae are viviparous, producing about 20 to 40 eggs based on the fertility data we obtained in 2016. In contrast to the reproductive strategies of invasive species that are territorial and oviparous and are more successful in producing up to 2,000 eggs in sexually mature females. We found here there is an important production of new fishes, about all the invasive ones, to colonize the new environment. A special mention should be made of the size of the introduced species, which go up to 15 or 20 centimeters, while in the case of Poecilopsis infans, his maximum size is about 5 centimeters, which is reflected in how the two species are the most important relation of fish biomass, with Lepomis macrochirus representing about 15% of the species found in the lake. Special mention, studies of stomach content allow us to observe the impact of the introduction of species as Lepomis on its direct competitors as Zogonithecus polypechus and Codea tripinis, which have suffered a constant decline, while the species that have persisted are those that avoid direct food competitions, as in the case of Poecilopsis infans, the most found species in the environment. Looking closely at the food ontology of Lepomis macrochirus, we can identify an omnivorous species with a tendency to eat insects, such as the case of as the genera Diptera, Decapoda, and Coleoptera, which is preferential article. We see in this graph of frequency that Diptera has a more important food item for Lepomis macrochirus, and it's more important in the largest sizes, where their consumption is associated with greater satiety and less hunting energy. In analysis of fauna, of benthic fauna, we also find that these articles are particularly limited in the oligotrophic environment of the lake, and where this represents a strong competition between all these species that have preference for consumption of insects, like we see Sogonitecus puripechus and Godea tripinis, that is also the species that are close to extinction. In the case of growth and the structure of the population, we can find that organisms are concentrated in 6 to 8 centimeter sizes, where in addition our gonadic maturation studies show that they were sexually apt to reproduce, which points out that it's a species with a strong tendency to continue growing. In this sense, the presence of females in higher proportions in the summer season tell us that the species maintains an unbalanced L15 to maintain its progressive growth in the environment. In addition, we make gonadosomatic index indicator that has a cycle of constant reproduction through the year, like we see in this graphic. At the same time, the condition factors, that is up of 1, are evidence of a species that is constantly expanding and has adapted environmentally to the new environment that they are colonized. But we want that this information became something real for the community, and we must to understand that in this case we must to find the alternatives of use of these invasive species. The fish obtained and not fixed in alcohol were dried to produce fluor, which was chemically characterized, and the results show that it's a fluor with high protein content. This leads to experiments as an organic fertilizer and as a food supplement for fish and the laboratory level. The information that I'm not presenting here, because it's part of the article we sent, indicates that this is a relative good organic fertilizer and that it's a supplementation proved to have beneficial effects on the growth of fish in experimental ponds. These alternatives of use are being taken to the community, to meetings with the guardians and the farmers of the region that have been taught that selective fishing of invasive species is positive and is something that they must do, as well as preparing sample classes for students and teachers on basic level about all in the region, so that they know the biological richness of which they are guardians. All this with the accompaniment of the local government. We also find that another problem facing our water resources is environmental degradation caused by pollution from human activities. Mexico is a country with an imminent industrial and agricultural vocation, reflected in the almost widespread use of many rivers as effluents from industrial waste, which has impact in the resilience of these ecosystems. To know the state of a climatic environment, that is to say with a unidirectional energy flow like the rivers, we usually use the recording of chemical parameters, which however do not represent in a punctual way really the state of a site, whereas these are highly dynamic and changing environments. For this reason, biological indicators have been used, such as benthic fauna, which through their life stories give a more reliable reading of change in time and space about in specific sites of the river. I will tell you about the study carried out in the river Duero, and in which I have participated in the last year. The river is located in the agricultural is used intensively in the agricultural industry of the region, principally in the west of Michoacán, to the city of Zamora, the biggest strawberry producers in the country. At the same time, its route joins the water of the Lake of Camecuaro, which is an important tourist area. It has an extension of 75 kilometers from its birth in the mountains of the Purépecha region to its mouth in the vicinity of the Lake of Chapala. In this work, the region was divided in 14 sample sites, which responds to sites identified as at points of good quality, like the head of the river and union of small channels of spring origin, as well as specific points of strong anthropogenic activity. In each of these sites, the environmental, physical, and chemical parameters were recorded and was made a characterization of the habitat, like, for example, identify the substrate of the river, adjacent plants, among other characteristics. Water samples were taken for microbiological determinations, and samples were taken from benthic organisms from each of the different microhabitats identified in each of these sites. One of the first things that we can observe as an environmental changes is in particular nitrogen compounds like nitrates and ammonia. Main interests are nitrates, which are sign of the use of agricultural chemicals, being the last sites like Capulín, Puente Cumuato, y Estanzuela. They are located in an area of a strong agricultural production, particularly affected by the increase of these nutrients, as we see in these graphics. We made a comparison between what we see in the last year and what we see 20 years ago. And we saw that these environmental parameters has changed so much that we can now make a comparison with what we see in the benthic fauna. In the case of dissolved oxygen, a parameter that indicates the capacity of the environment to support life and carry out oxidate reduction reactions, we observe that since the river enters the urban and agricultural area, its values drop. For example, later of Central de Abastos in Zamora, we found that the oxygen is close to zero. And that really points out that the kind of degradation in the environment and how in these environments can be life that when they don't have oxygen. This data was contrasted with the diversity and richness of benthic species, which also begin to decrease when they entering the sites of a strong environmental impact. In the case of how the biological communities are structured, we found through the use of indices such as the relative importance that the species considered as decomposing are more abundant, like Lumbriculidae and Chironomidae and Absogammaridae. We see that these species considered as decomposing are more abundant in the whole of the river, which indicates that this is indeed a river in process of accelerated eutrophication, and where the role of these organisms is key to maintaining environmental balance. The index of water quality are quite illustrative of the process of degradation of the river, where we can see in the map from its head to this mouth, its degradation is constant. From excellent sites in the immediate born of the river, and be degraded to the mouth and the vicinity of Lake Chapala. In this sense, is for the comparison with what was observed only a decade ago. We have information of the state of the river make a decade ago, and we make a comparison of what we saw now in 2019 with what we see in 2009. And the level and pace in which the degradation of this environment has developed is acute. As for what we see in habitat quality, there is a correlation between maintaining a healthy habitat with the physicochemical parameters of water, finding the sites near the entry of spring water. This is the integrity of habitat index, and we find that the optimum sites are the beginning of the river and some places where water from little rivers that confluent in this big one are part of help the restructuring to really maintain the habitat for the for the benthic fauna. Finding that the sites diversity indices, we make the indices of Shannon, also show us that as our water quality has degraded, the richness and abundance of organisms has suffered, having today generally less diversity, for example, in the beginning of the river. And that is to say, we went from levels of 3, that is considered to say of good to excellent quality, up to degraded sites with fragmented habitats, like we saw in Estanzuela and Capulin. The BMWP, there is an index to know the health of the environment, are also revealing or to identify an obvious problem of an environmental losing its resilience capability. We now compare what we saw in the 80s to what we see the last year, and we find that most of the sites are belong of the state of ecological resilience we saw 20 years ago. With data obtained from environmental, physicochemical, and biological parameters, major component analyses were performed, which grouped the sites according to what we could observe in the 2 indices, finding that sites considered to be in degradation are sites with high biochemical oxygen demand, turbidity, nitrates, and a presence of bacteria related to anthropogenic activities. As well as that good quality sites are sites with total transparency and high levels of dissolved oxygen. These sites that are classified as the better ones, are sites with long levels of oxygen who leads to a major degradation of the organic contamination of the water. Following, we make a distance analysis, and we could find that benthic organisms are grouped with certain environmental parameters, finding that greater diversity is directly proportional to parameters such as abundant oxygen and unstable pH. But how we can convey this information to real actions with the community? In that sense, we have encountered some problems that contribute to the actual state of the river. First, the use of a large quantity of chemicals to protect the production of strawberries in the region, as well as the loss of activities such as fishing, which has been abandoned by the habitats of the vicinity of the river and the lake to the lack of stability in the water of the lake. Because in dry times, the lake is dry. In that in that moment, in the dry season, we see a bloom of water lily. That is an invasive species compromising the health of the ecosystem and the stability of water resource by accelerating evaporation and increasing zones of hypoxia. The work that continues is to establish communication channels with ejido, municipal, and state authorities to establish measures to recover this important river. Now I want to tell you about that last year I have changed my field of research. Understanding the new frontier of ecology of conservation is now in molecular biology. One of the main questions in evolutionary biology is the role of place, the genetic regulation in the emergence of new phenotypes. In this order of ideas, Darwin himself has considered the role of the role of environmental adaptation in the speciation process. And one of the most controversial modern theories postulates by Kimura that a set of neutral mutations become fixed in a population when they give an adaptive advantage and during a genetic drift. The process of expression and repression of genes in a given time and space are essential for cell and its order to process in so-called transcription circuits, formed by proteins known as transcription factors, who recognize a sequence known as cis regulatory regions. Recognize this sequence some enzymes and cofactors are recruited and carry out the expression of a gene. This is the translation. These circuits undergo constant modifications in a normal way, derived from mutation characteristics of the replication process, which leads to want to know how these changes lead to emergence of a new phenotype. To know this, we use model organisms where the yeast, like the yeast, due to their easy genetic handling, the possibility to work with the species at different evolutionary distance is particularly used. In my case, I currently work with a species of the genus Candida group in the clade CTG, which translates this codon for setting instead of leucine, that is the normal. And that despite being environmentally closed, they have colonized divergent environments like Candida albicans, that is a pathogen of the humans. In the laboratory, we focus on the study of phenotypes such as the formation of biofilm, and we want to understand how the adaptation to different ecological niches has impact on changes in the expression of this phenotype and an important process of the member of this clade and its pathogenicity, for example. As a conclusion, we know that it's essential to know the integrity of environment to propose ideas for management and prophylactic ones. To control invasive species, it's necessary to know the biological characteristics and the environmental conditions, and that make us possible establish ideas of control and exploitation. And if we want to this idea of conservation, we cannot ignore that now the future of this ecology is now based in understand the biology and adaptation process. I want to say thank you to my lab mates in Laboratory of Aquatic Ecology, Aquaculture and Fishing in Sidir, Michoacán, particularly to my friend Guadalupe Cabezas and Paulina Victoria, who they are the woman leading now the project in La ALBERCA, and my friend Guillermo Magallón, who is the lead of the research in the Duero River. And of course, my mentors in the master degree and degree thesis, Rodrigo Moncayo and Carlos Escalera, and now my advisor in the PhD, Eugenio Mancera at Sinvestaff. And that's everything. Thank you.
Okay. Well, thank you very much, Marco, for your presentation. Well, now we have time for questions. So people, if you have any questions, please let us know here. Ahora estamos aceptando preguntas en portugués, o en español, o en inglés. Aí fica na vontade. Dá um tempinho. I think it's very interesting how you're using some different approaches to tackle this problem. Like, I don't know.
I want to tell something. When we work in Rio Duero, we found that this river is dynamic. It's a dynamic environment, but the velocity of flow it's not very fast. And we worked before in another river called Cupatizio here in Michoacán, and we found that this river has it's more contaminated, but there is a much more level of oxygen, and that allowed the river to maintain his resilience to his burn, to his disappearance. And my advisor of master degree say that in this moment it's important to understand that the dynamic of the river is essential to maintain his resilience. And we saw in the Duero that is a lake with so much management for agriculture and for the urban uses, that is the main problem to the environment resilience of this environment.
Well, yeah, that sounds like a pickle. Well, good luck with that. It seems that we don't have questions, so I think this is the end of the EcoEncontros. Well, thank you very much, Michael, again, and thank you for all the people that were streaming this presentation. So till the next one. No. Chao, chao.
Creo que mandaron una pregunta.
Espera ahí. Una pregunta. This is thank you, Mark. There's some people saying thank you. Okay, yes, we have one question. The question is, do you think these anthropic impacts are specific to this lake, or they're more general in Mexico?
This lake, La Alberca, it's a lake with some special characteristics. The first one is that we have geothermal activity, we have high levels of salinity, and we have a turbidity that is about 16 centimeters, who made it a lake with oligotrophic characteristics. This lake has special characteristics, and for that, the lobina, that was the species that originally wants to be explored in the lake, can be successful in this environment. The thing is that Lepomis and Copterendali belong to a family, the Cichlidae family, that are fishes with a plastic environment that really give us the chance to adapt to this new environment. So no, the impacts that we describe in this lake are particularly of this environment and not is a general of what happened in Mexico. My master advisor, Carlos Escalera, and Rodrigo Moncayo has a lot of information about invasive species in Mexico. If you want to see some of his articles and send them a message, they are very kind person, and they absolutely answer your question.
Okay, any more questions? Okay, Marco, I think that'll be it. Thank you very much again for your presentation. It was very interesting, and good luck with your PhD.
Thank you. La voy a necesitar. Muchas gracias.
Sí, yo estoy en la misma. Bueno, com muito gusto. Para toda a galera, muito obrigado por ter sido parte mais uma vez de um EcoEncontros. E até a próxima semana. Um abraço a todo mundo e se cuidam aí. Tchau, tchau. Abraço. Tchau, Marco.